Katie's brilliant talk. We have Renier Brentjens from New York. He trained in Buffalo with an MD, PhD in microbiology, making his way all the way to New York at the MSK. And he's been focusing on CAR T-cell therapy, pioneered CLL therapy, ALL therapy, and we'll speak now about next wave of CAR T-cells that we may see in the future.
And probably should point out that I just recently moved back to Roswell Park in Buffalo. So it wouldn't make any other sense. So I want to thank the reviewers. I want to thank the reviewers for inviting me. This has been a phenomenal meeting, and it's been a pleasure and an honor to be part of that. Here are my disclosures. So one of the lousy things of being day 2 at a CAR T-cell meeting is that your introductory slides have become extremely redundant. So I think I'll move on from this slide. But this is actually a slide that I made probably 15 years ago, and it still remains quite relevant. This is early work when I was working in Michel Satterline's lab to look at a first generation of CAR T cells and see whether we could actually demonstrate that these CAR T cells could kill tumor cells, not just in a petri dish, but also in a mouse model. And so we used skid beige mice model mice. You could see here we could monitor expression of the CAR T cell. We could show specificity in vitro, and we could expand these cells in the context of at the time our favorite cytokine IL-15 to what we thought would be clinically relevant numbers. And I think, if I'm not mistaken, this is the first published indication that we could eradicate systemic disease with CD19 targeted CAR T cells that were similarly infused systemically a week later. You could see here with the first generation CAR that just contained a ZD3 zeta chain, we could eradicate disease in anywhere from 50 to 100% of the treated mice. When we got a little cocky and we decided we would try to treat other B-cell tumors before we would submit the manuscript, we started running into trouble. And we ran into trouble very early on, ironically, with NALM-16, which is a B-cell ALL tumor cell line. And back in those days, still utilizing xenotransplant tumor models, we hypothesized that this might have been due to the fact that there was a lack of signal 2 or co-stimulation, where the Raji tumor cells would provide, because they were CD80 and 86 positive, or at least 80 positive, NALM-6 obviously derived from a more immature B cell, did not express any co-stimulatory surface molecules. So we engineered the tumor cells to express CD80, and lo and behold, we were able to get long-term survivors when previously we were unable to do so. And that led to a single slide that represents about 3 years of my career in the laboratory without a single sentence, where we started looking at how, we and others started looking at how we could generate a more potent CAR design. And so these second generation CARs, which have the CD28 or the 41BB co-stimulatory signaling domains, these are the ones that are now FDA approved. And for most investigators, this is kind of the stepping off point. Third generation is also called, with multiple co-stimulatory signaling domains, have not yet seen, I think, as much promise or been FDA approved in the clinical setting. In our hands, the level of CAR expression when the gene gets larger went down. And there's this concept that there may be a Goldilocks effects where too much co-stimulation may not be good for the T cell either. So we repeated our initial experiment where we couldn't eradicate disease in our xenotransplant model, and we now could do much better with a second generation CAR, which we termed 1928 zeta because it had the 28 co-stimulatory domain. And this led to myself and Marco Davila, now it was 2009 when we opened up our first trial with adults with relapsed and refractory ALL. We published the first 5 patients. These are, again, adult patients. Several patients had morphologically prevalent disease, 60–70% blast. And by deep sequencing analysis, we could demonstrate that in 5 out of the first 5 patients that we treated on this protocol, we got MRD negative remissions. And overall, and again, I hasten to note that this is not an intent to treat analysis, but overall, the patients that were treated and did receive CAR T cells, 85% achieved a CR, of which 2/3 were MRD negative, either by high sensitivity flow cytometry or by deep sequencing analysis. As pointed out, and what has become very clear, remissions are not as durable as we would like. Even though the initial remission rate is really quite high, relapses are quite common, especially in patients that actually have morphologic disease, which we defined as greater than 5% blasts in the bone marrow. And so this leaves room for improvement. And similar results were noted in multi-center clinical trials. This is derivative work from the University of Pennsylvania, the Novartis trial, and this was the first FDA-approved drug for ALL. This is in the context of pediatric patients. And again, you see, unfortunately, that while initial remissions were quite high, the relapse rate was significant as well, demonstrating that the CAR T cells, as it is a product right now, is not the end product. And so there is room for a significant amount of room for novel innovation, as well as to improve these long-term outcomes and make remissions more durable. And what I'd like to do is kind of show you some vignettes of how we think of how we can improve CAR T cell function and illustrate some other lessons that we've learned in the process. The first thing I would like to mention along the same lines, Eric Smith in the lab, Sham Malankody in the clinic have worked for multiple years identifying targets for multiple myeloma. We, like many others, developed BCMA-targeted CAR T cells that have been tested in the clinical setting. But Eric actually identified several other very interesting targets, and GPRC5D was one of those targets that's also highly expressed on multiple myeloma. And again, it goes back to addressing this issue that we have, which is similar with CD19 CAR T cells, initially high response rates, but unfortunately a significant number, even these are the two commercially available CAR T cells for BCMA. And in both settings, you can see that there's a lack of durability in response rates. So we identified, or Eric rather, identified GPRC5D, which is expressed in the skin and in the nails, but is significantly expressed on multiple myeloma tumor cells. And Eric did a very interesting experiment, or at least a very telling one. Mice were injected with two populations of multiple myeloma cell line. This is all done in the xenotransplant setting, knocking out BCMA in one, and the second expressing GPRC5D and BCMA universally. You can see here when you treat with a BCMA-targeted CAR T cell, you initially get regression of tumor, and then tumor relapses. And if you then go back and try to treat with BCMA again, you get tumor expansion due to immune escape. But if you go after GPRC5D, you again get these mice back into remission because, again, dual targeting shows promise. We have recently completed a phase 1 study at Memorial treating patients with relapsed and refractory multiple myeloma with GPRC5D targeted CAR T cells. I would note that the overall response rate was 81%, but the CR rate, especially at the higher doses, was up to 60%. The significant thing, which I made sure that Sham decided, we made sure that we treated patients that had previously failed BCMA-targeted CAR T cell treatment. And as you can see here, a significant proportion of those patients would still respond to the GPRC5D CAR T cell. And again, this is the survival plots. The follow-up is still relatively short, with a median of 17.9 months. But as you can see, there seems to be a dose response, indicating that this may be a promising second target to go after. More significantly, we are about to embark at Sloan in a second trial wherein patients get equal doses of BCMA and GPRC5D targeted CAR T cells, which I think is one of the first of its kind of true dual targeting in the setting, in this case, of multiple myeloma, given the fact that there are these two attractive targets to go after. And again, it illustrates this idea that dual targeting, which has been a subject of intense scrutiny and interest, may now at least start to be realized in the clinical setting. So what are the questions that we still have to ask about CAR T cells? Well, there are many, including what is the etiology of differential responses to CAR T cell therapy? We've illustrated that bulky disease tends to, even though the response rates are the same, the relapse rates are higher. And what is the role of bulky disease? What is the optimal clinical setting to treat patients? More significantly, it's been brought up by several speakers here, is what is the role of the hostile tumor microenvironment and CAR T cell function? The follow-up question to that is, how do we build a better T cell, and can we extrapolate this technology to solid tumors? And again, it's been mentioned now by multiple of the speakers here that there are a multitude of factors that could inhibit CAR T cell function, especially and likely much more so in the setting of solid tumors. The first of which is, again, tumor antigen escape. The heterogeneity of targets in solid tumors is much more diverse than it is with, say, CD19 or BCMA, where virtually by flow cytometric analysis, 100% of the tumor cells express, at least at the start of treatment, express the target antigen. There are inhibitory cytokines, there's immune checkpoints, regulatory T cells, which have been discussed, as well as myeloid-derived suppressor cells, tumor-associated macrophages, and cancer-associated fibroblasts. And I wish this was a complete list. It isn't. And so if there are those of you that think that there are other things to add to the list, including the pH and nutritional status within the tumor, obviously. So some of you have probably seen this slide before. I've grown very fond of it. So what we really have in the CAR T cell world right now is kind of a proof of principle. We have a Model T Ford, and what we're really looking for is a Ford Mustang. And again, I bring that up because I actually have that car with a soft top and obviously a stick for those of you, which is about 100 more horsepower than Marcel's Porsche has, but that's something for another discussion. And so we came up with this concept that we termed, and actually one of my technicians in the lab called me up a year after he had left for industry and said, I got the name, and armored cars. And so we started using this term armored car, and we define an armored car as one that has a chimeric antigen receptor, but is additionally engineered with additional genes to, for example, express pro-inflammatory cytokines, co-stimulatory ligands, or secretable single fragment length scFvs, which, again, I'll give examples of which, some of which have published, some of which hasn't been published. But that gives the concept here is not so much that the CAR T cell is a killer, but that the CAR T cell is kind of a troop transport mechanism that can infiltrate into the tumor by virtue of the chimeric receptor, and then can significantly modulate that tumor microenvironment, which we find is really quite inhibitory. Now, the examples I'm going to give all are mechanistically divergent. They don't all function the same way. And what this implies is, at least from our standpoint in our lab, we believe that ultimately you biopsy the tumor, you assess the tumor microenvironment, and then you pick out what the best armors are to be able to address that particular tumor for that particular patient. So the first example is IL-18 secreting CAR T cells. This was work done by Amaro Avanzi, who was in the lab at the time, a postdoc in the lab at the time. This is actually follow-up to IL-12, which was our first cytokine that we engineered into T cells and have published many moons ago. Here we utilize a fully murine system wherein the normal B cells as well as the tumor cells in this syngeneic immune competent mouse expressed human CD19. We used our clinical CAR, and then we used a P2A element to have the T cell secrete murine IL-18. Again, as I said, it was a transgenic model. It's immune competent. We did not give any preconditioning chemotherapy in this model. And as you can see here, when you just treat these mice with a CAR targeted to CD19, there's no survival benefit. If you have an irrelevant CAR but IL-18, there's no survival benefit. But when the CAR is targeted to CD19, secretes IL-18, you get 100% long-term survival. And we were really impressed with this result because this truly fit the definition of synergy. And so we wanted to look to see what the mechanism behind it was. We could tell in blue here are the armored CARs. You could see that persistence was far greater than what could be detected with a CAR alone T cell. When we looked at B-cell aplasia, since the normal B cells express human CD19 in this model, we could see that relative B-cell aplasias were evident out to 150 days. And again, you could see this by flow cytometric analysis. And out at 150 days, we took bone marrow from these mice, predicting that there would still be CAR T cells detectable. We did PCR analysis. And as you can see, out to 150 days, you could still detect the CAR T cells in the bone marrow. We also tried to see whether these mice would, with this persistence, would be resistant to tumor rechallenge. And so we rechallenged these long-term surviving mice. And as you can see, about 50% of these mice were able to resist tumor rechallenge with the CD19 positive tumor. We then started asking, what else? Is there any evidence that this IL-18 can help recruit endogenous immune effectors? And we did that. There's a variety of studies that are in the manuscript, but I don't have time to present, including the fact that it modulates M2 macrophages to an M1 phenotype at the site of the tumor. There's an increased number of NK cells at the site of the tumor. But just to demonstrate that this would have a spreading, or that the endogenous immune system could actually have effect, we injected these mice with a combination of target antigen positive and target antigen negative tumors, and then treated them with the CAR T cells. And as you can see here, about 25% of these mice are long-term survivors, despite the fact that even at the start, half of the mice did not express the target antigen. ELISpot analysis further confirmed, even against CD19 negative tumors, that there was an increase of endogenous T cells that recognized the tumor. Again, this epitope spreading effect that Malcolm was talking about yesterday. Now, it's going to seem like I'm shifting a little bit, but bear with me. In another project, Janneke Jaspers from in my lab started looking at another target, DLL3. This is expressed on a lot of neuroendocrine tumors, including small cell lung cancer. Clinical experience in the past dealt with humanized antibodies. The ROVA-T was tested clinically. High payload-related toxicities was an ADC. But as you can see here, it's an attractive, nonetheless a very attractive target, especially in a disease where therapeutic options are really quite limited and outcomes are really quite poor. And this is the point of this slide, is we then utilized a phage display, human scFv phage display library. Back in the old days when we started doing this, we would actually only have a handful of hybridomas that we could assess for our CAR T cells. We were very fortunate. And the field was that the two hybridomas, the FC63, FMC63, that's from the ATC, and the SJ25C1 that we had, both of those are scFvs that function well with CARs, but now we have the luxury of being able to look at panels, 20, 30, 40 different binders. Janneke picked out 3. You can see the affinity is markedly different between the different binders, and you can also isolate where it binds to, with the idea now that more proximal binding, and certainly in the setting of certain antigens, is more favorable and have better outcomes. Janneke assessed these 3 further from a panel of 20 to 30. You could demonstrate that there was efficacy in vitro. And you could also demonstrate that all 3 of these that she selected had efficacy in vivo, but it was somewhat modest and somewhat disappointing. And so having the tools that we had available, we repeated those studies, in this case with one of the constructs, the 2J8, and we combined that again with IL-18, and now we get far more robust long-term survival in this model of small cell lung cancer. This construct is going to move forward to the clinical setting. We're in the process of doing that. Another approach that we looked at, not using cytokines, and again, it's a platform, right? There are multitudes of cytokines, and if there are those of you in the audience saying, well, I got a better cytokine than IL-18, God bless. We started with 12. We think 18 is better than 12, and we're actually looking at the combination of both as well. But another approach would be to constitutively express ligands such as CD40 ligand. This was work done by Nicholas Kuhn, who's a graduate student in my lab, and looked at the idea that CD40 ligand could not only modulate the tumor cell itself. It's been long since reported that in some tumor cells it upregulates CD80 and 86, but more significantly, the interaction of a constitutively expressing CD40 ligand CAR T cell with a dendritic cell could stimulate secretion of IL-12, but also cause cross-presentation, and we would predict that, again, we would get epitope spreading in that setting as well. So Nick, again, utilized in immune-competent A20 tumor model with no lymphodepleting chemotherapy. Looked at two different models, one in BALB-C, that's the A20, and the ALL1 model in black 6 mice. And as you can see, with the absence of lymphodepleting chemotherapy, we still got long-term survival in both settings, which you wouldn't get in the context of CAR T cells that are not armored with CD40 ligand. We looked at the different aspects of why this was happening, and again, I'm just giving a brief synopsis, but we looked at dendritic cells in the tumor to see if there was evidence of maturation. There was none, which we were quite surprised. But Nick also looked in the spleen, and here you could actually see evidence of dendritic cell maturation occurring in lymphoid tissues. And as you can see, as well, upregulation, cytoplasmic staining for IL-12, which was observed here, again, demonstrating that there was maturation of dendritic cells, not necessarily at the site of the tumor, but certainly in the site within the spleen. To then look to see what was happening, could we tell whether there were endogenous effector T cells along with the CAR T cells that were being activated? And so we, again, looked at activation markers, and as you can see here, the CAR T cells, when they are armored, have a significant, in red, have a significant increase in activated T cells in the tumor, and you could see in CAR-negative T cells, so T cells that are endogenous, also upregulation when compared to CAR-alone T cells, upregulation of activation, and that's seen again, both in the spleen as well as now within the tumor, consistent with this concept that epitope spreading was occurring. And to further validate that, we put into Thigh 1.2 mice, we injected Thigh 1.1 CAR T cells, and we could sort the 1.2 endogenous T cells. And again, by ELISpot analysis, we could demonstrate that these endogenous T cells now recognize the A20 tumor. And if that wasn't enough, reviewer 3 still complained. We further validated by looking at re-challenging those long-term surviving mice with tumor that has the CD19 knocked out. And in both cases, again, you see almost 100% long-term survival, consistent with recruitment of the endogenous immune system, likely mediated through definitely mediated through dendritic cell maturation. Ooh, I'm going way too fast. There's going to be a lot of time for questions. So then a third platform that we've utilized, that we've looked at in the laboratory, is whether we could armor CARs with single fragment length antibodies. And we initially, to be honest, looked at CD47. That was our initial target, but that story got a bit more complicated with the need for FC binding with the macrophages. So we initially went with PD-1. We generated a series of human PD-1 binding scFvs, and the idea there was that a CAR T cell could secrete PD-1 scFv that would protect itself, but in addition would also have a not just cis, but also in trans, be able to protect tumor infiltrating lymphocytes. The idea here, again, is that the CAR T cell, by virtue of the CAR, locates itself to the site of the tumor. There it secretes the payload, and therefore we could minimize the toxicities that are seen with systemic either cytokine infusions or in this case immune checkpoint inhibition. So in a very simple initial experiment that Holly Pigram at the time in the lab conducted, Holly took mouse fibroblasts that were engineered to express high levels of PD-L1, and then took T cells that secreted this E27, which is the humanized scFv that binds to human PD-1. And as you can see here, if you just take CAR T cells that you grow on PD-L1 expressing fibroblasts, you can see there's a significant abrogation of proliferation. These arrows represent restimulations. And if you do the same experiment on empty CAR T cells, so these are activated T cells, on empty fibroblasts, they grow. Now if you take the same PD-L1 expressing fibroblasts and you take activated CAR T cells that can block PD-1, you can see there's restitution or resolution of growth inhibition. And PD-1 not only can inhibit the proliferation, but also cytotoxic activity. So flow cytometry analysis at 24 hours of a co-culture of tumor cells, in this case, NALM6, the ALL cell line that we mentioned earlier, that were engineered to express high levels of PD-L1. You can see at 24 hours, the tumor is outgrowing the T cells significantly. When those same CAR T cells now secrete this E27 PD-1 blocking scFv, there's resolution, CAR T cells are activated, proliferating, and are able to eradicate tumor cells. We expanded this work to the in vivo setting, again, engineering Raji tumors that express high levels of PD-L1. And as you can see here, the PD-1 scFv secreting CAR T cells have enhanced anti-tumor efficacy in this xenotransplant model, as they do with NALM6 tumor cells that have also been engineered to express high levels of PD-L1. We expanded this to solid tumors, which is really the area where we think it's going to have most significance. So we took SKOV3, this is an ovarian tumor cell line. Again, this is in a xenotransplant model. Intraperitoneal injection, we injected CAR T cells, also intraperitoneally targeted to MUC16, which is the 4H11, is what the 4H11 CAR recognizes. And as you can see here, when you compare to CAR alone, the survival significantly increased when the CAR T cell also secretes this PD-1 binding scFv. We further wanted to demonstrate that this phenomenon could be seen both in cis and in trans. So in this follow-up experiment, we did the same experiment, but this time the tumor-targeted CAR T cells did not secrete the scFv, but rather we injected another population of CAR T cells that were specific to CD19 and secreted the scFv. And again, you could see that there is a trans, or that those cells that were not tumor-specific were able to rescue tumor-specific T cells targeted to MUC16. And then finally, we did this in a syngeneic immune-competent model, again, intraperitoneal ID8 ovarian tumor cells, CAR T cells, in this case mouse CAR T cells, either engineered to secrete the RMP114 scFv that Jim Allison provided for us, or did it with a direct comparison with CAR T cells in combination with infusion of the RMP114 antibody. As you can see here, the survival rates were very much the same. And this, we could demonstrate, led to a longer persistence of these CAR T cells in vivo, again in this immune-competent syngeneic mouse model. So here the armored CARs can still be detected by PCR analysis, as well as CAR T cells in combination with systemic PD-1 blockade, while CAR T cells alone were no longer detectable. And again, to further validate that, we took long-term surviving mice, rechallenged them, and could demonstrate there was still enhanced capacity of these T cells to eradicate tumor upon rechallenge. And so my conclusions are that there are promising outcomes with CD19 and BCMA CAR T cells that serve as a proof of principle that this approach to adoptive T cell, that this is a valid approach to adoptive T cell therapies of cancer. There remain significant limitations in this technology, not only in the context of heme malignancies where the relapse rates are high, but even more so in the context of solid tumor malignancies where there is a much more suppressive tumor microenvironment. There is antigen target heterogeneity, which we don't see as much in the hematologic malignancies, and so on. Additional genetic modification of CAR T cells, the ceiling is really quite high. I've shown you at least 3 examples where additional engineering in both xenotransplant as well as syngeneic immune-competent mouse models enhances survival. Given the fact that the mechanisms and the pathways differ between those, we've not even yet started, but we plan to start combining those different approaches, hopefully in more valid syngeneic immune-competent models. I think that the most difficult aspect of that I have actually noted, and one of the reasons why I moved to Roswell Park, is that rapid translation of these approaches are absolutely required in phase 1 clinical trials for this field to move forward at the pace that it possibly can. The innovation is there. There are multitudes of happy mice that have survived because of these new innovations, and unfortunately, we need to translate that to the clinical setting and to our patients. This is still a photo from the lab at Sloan. I haven't had time yet to take a new picture at Roswell. Thank you.